Before describing the article, Deciphering the Rhizosphere Microbiome for Disease-Suppressive Bacteria, I'd first like to introduce the pathogen which this study is based upon and the reason for this investigation, Rhizosphere solani. R. solani is a soil-borne fungal pathogen that attacks the roots of important agricultural crops such as sugar beet, rice, and potato, causing significant economic losses, particularly its seedlings. A new trend in plant disease management is to increase disease suppressiveness by manipulating the microbiome around the rhizosphere. The rhizosphere is the area of soil directly adjacent to the plant root that is affected by plant secretions. Disease-suppressive soils are soil which susceptible crops do not suffer economic losses despite the presence of the pathogen. The opposite is a conducive soil, which allows the disease to progress uninhibited. The aim of the study was to identify the bacteria and the genes associated with the soil that is suppressive to R. solani. A soil suppressive to R. solani was discovered in the Netherlands. The authors then prepared 5 treatments of the soil: suppressive, conducive, a conducive soil amended with 10% suppressive soil, and soils heat treated at 50 and 80 degrees Celsius. So this graph shows that in the amended soil, some of the suppressive properties are transferred, and in the heat-treated soils, suppressiveness is completely or partially lost, proving that suppression is biological. They then also prepared a suppressive soil inoculated with R. solani to identify any taxa that responded to the presence of the pathogen. The authors then used phylochip analysis to identify the important bacteria in each of these treatments. They then compared the differences between the soil treatments to show the bacteria and their abundance associated with the suppressive soil. This analysis revealed that beta proteobacteria was the most important bacterial group associated with soil suppression, and focused further their work on Pseudomonads, a family within beta proteobacteria, which are already known to be suppressive of other fungal pathogens. Pseudomonad isolates were taken from the suppressive soil and grown in selected media. The ability of the isolates to suppress R. solani was then tested. The most effective bacteria was then mutated so that it no longer functioned, and genetic sequencing was used to identify which genes in the Pseudomonad bacteria were important for the suppression of R. solani. In this study, the authors achieved their aims of identifying the important groups of bacteria involved in suppression, isolating the most important bacteria, and finally determining the important genes in disease suppression. However, the authors suggested that because Pseudomonads were not the only family of bacteria associated with suppression, that they believed that disease suppression was more likely to be due to a consortium of bacteria with different genes.